One, go.
Hello everyone. Welcome to the first English presentation at Hemmaodlat. My name is Enrique, as you all know. I'll be talking about anthroponics. We don't have really a big projector, so you'll have to bear with me and look at the screen here or whatever I write in the board. Let us start. I took the time to draw a short evolution of the way we produce food since prehistoric times to nowadays. So on the left side, you have what we call soil farming, for example, or geoponics, and also fishing. And then as we progressively go forward, we evolve into the other types of growing that now we discuss here at Hemmaodlat, such as hydroponics, aquaponics, and now anthroponics. All of you already know more or less what hydroponics and aquaponics means. Anthroponics basically has the prefix anthropo, which means human, and the ponics part, which means to work. So we put the human to work literally. However, online, most people refer to what we call anthroponics as ponics. In here I divided, you can see here, there's actually several types of potential anthroponic systems. There's ponics, there's ponics, and there's the combined one. Nobody, as far as I'm concerned and have read about, has done the other two. So we're just now starting to discover the first one, and maybe in the future we'll be able to do the other one. So anthroponics, how would we define anthroponics? I would define anthroponics as a recirculating soilless agricultural system that uses bacterial cycles to convert human biowaste, such as urine, into plant fertilizer. So if you're familiar with aquaponics, we take the fish waste and we convert it to plant fertilizer. We do the same with anthroponics, except we don't use fish, we use human waste. So this is just to give you a little idea of how we are in the terms of growing systems and what we can do in the future. But a more important question is why we're deciding to investigate anthroponics. From what I was able to gather and read, it just doesn't make sense nowadays to treat the waste as we do. What we do when we go to our toilet is we do our type 1 or type 2. We dilute it a lot. It travels a lot of kilometers to a big central system, and then we spend a lot of energy and materials separating everything again. This is why separation of waste at its source is becoming more popular, especially in Sweden, with toilets which have different types of waste collection. But more importantly, we have the two big problems with why we want to invest in anthroponics, which is the environmental concerns and the economic concerns. So environmentally, it doesn't make sense for us to have this line production of we consume, we use it, a resource, and then we just waste it into the environment. We're also wasting fresh water, and we use a lot of energy in this whole process. Concerning how other sources of soilless grow might compare with anthroponics, for example, with hydroponics, you have to buy the minerals in the form of the bottle, and they have to come from somewhere. Usually they are mined at a location, and then they are mixed into the solution that we buy and use here at Hemmaodlat, for example. For example, in aquaculture, they have to produce the fish feed, and that fish feed, which includes fish protein, has to be fished from the ocean. So that there's an environmental concern there, because even though we might shift our aquaculture production inland, where it's more sustainable, we're still fishing to feed the fish that we want to grow here. On the other hand, it's just expensive to buy the mineral bottles that we use, for example, here, as well as the fish food. Now, to go a bit into more detail as to how it works, don't get scared. It's quite simple once you get into it. But the main thing you need to know is that urine, if this represents urine, 95% is made of water. So it's already a lot diluted. And this should make you think about how I mentioned earlier that we dilute it even further, and that just complicates when we want to separate the nutrients again. The other 5%, half of that is urea. So that's about 2% urea. And the other half of that 5% is the soft ions such as chlorine, potassium, sodium, and phosphorus and other nutrients. There's also some organic nutrients which are harder to break down, but they're not very important for what I'll be discussing today. So basically, we take urine, we collect it, and then we sterilize it. Now, some authors recognize that urine is already sterile, particularly in males. In females, for example, we have the issue that, well, that's not what I meant actually. That came out wrong. With urine, we have to sterilize because of potential bacteria problems. Concerning the method of which we sterilize, we have to sterilize it through a process known as ammonia volatilization to urea, from urea. So this means that in this equation over here, the first one, we have our urea, and then through a natural process, which is we just place the urine in a closed container, it converts itself into carbamic acid and then into ammonia. Now, the reason I mentioned females before was the fact that depending on how urine is used, if we want to use it in a hydroponic setting, some females use hormone control, and at the moment there is no way to treat that in both in a normal wastewater setting as well as here. So for now, when we talk about anthroponics, we restrict it to a male use, for example. So that's about it. So when this first process occurs, that we convert the urea to ammonia, the pH of the solution increases to 9. This is how the solution is sterilized. Bacteria that might be harmful to us cannot survive this high alkalinity environment. So this is the first step that we do. And then we pass this ammonia solution through a biofilter. A biofilter contains beneficial bacteria that we grow through, and they convert through a process known as nitrification the ammonia or ammonium. The ammonia or ammonium is just a different form of ammonia that switches between one or the other depending on the pH of the solution. So then we have the ammonium that passes through the biofilter, through the process known as nitrification, and these two group of bacteria over here, or over here when you see it convert. So they convert it into a plant fertilizer, nitrates, first into nitrites and then into nitrates. The plants then uptake those nitrates and other compounds from the urine, and the water is cleaned. So usually when we talk about anthroponics, we talk about a recirculating system. We add the urine as the nutrient source, and then the water is just recirculated until it's cleaned. And then we add a bit more to keep the plants growing. So this is overall how it works. It's very similar to aquaponics in concept, except that instead of using the fish waste, we use the urine. A bit more about the history now. I keep talking to aquaponics, back to aquaponics, because it's very important. Aquaponics practitioners were the first ones that started experimenting with this. One such case is the American woman Alice Landis. Now, Alice noticed it that some aquaponic people, when they're preparing their system, they add ammonia to prepare the bacterial colony. These bacteria need a source of ammonia to develop first. Some people use fish that might die during the process to inoculate the system with the bacteria. Other people use ammoniac directly into the system. Other people add food fish. But some aquaponic practitioners used urine. So she was really the pioneer here, because back in 2007, she decided to run a whole system just with her urine. So she did what I explained before. She kept the urine in a sealed container until the pH reached a level of 9. And she was quite thorough in experimenting and measuring all the levels of different nutrients to see how it evolved through her system. Her system is a type of system that we haven't talked here at Hemmaodlat yet. It's called the barrelponics system. It's an open source system that uses a couple of blue barrels cut into several shapes and then connected through a constant flow. But you'll be able to see here, this is the top of her system. She used as a media expanded shale, which is a very porous and light media and material. As far as I was able to tell, she used that little liner there to protect the sump, or the lowest part of the system, from any light, so that algae wouldn't grow in the system and tamper with her results. But as you can tell, she had already pretty good growth of herbs and greens. I read also somewhere else that she experimented later on with some fruits, such as tomatoes, and she had equally good success. And overall, her experiment was a success, and that's what would eventually become part of my thesis. This is what inspired me to eventually go and try to develop this further. And that's when my thesis comes in. So back in 2012, I arrived in Sweden to do my thesis, initially just in aquaponics, but then alongside Louise Lundberg, we decided to do a trial with anthroponics. At the time, we referred to it as peaponics. The trial lasted a good deal of months, about 6 or 7 months. It was in Skåne, and it might look like a very complicated system at first, but all that you really need to know is that here is the lowest part of the system where we added the nutrients. Then the water is pumped both to the top of all the towers here and to this system that has a flood and drain, or ebb and flow system. On the other hand, there is a floating raft right over here, similar to a DWC, or deep water culture system. The initial goal was for us to build several aquaponic systems and one anthroponic system, and compare how would the water quality change over time, and can we compare it with how aquaculture wastewater looks like and how source separated urine that has been treated looks like. So that was the goal, and this is some pictures of what we built before and after. We really did not have many resources, so we tried to use whatever she had at her farm, and we recycled and upcycled a lot. And you really get a sense of the scale of the system here. This was only powered by her urine. So about once a week, she added what one person would add during the day, and we grew a lot of food. We grew cucumbers, tomatoes, strawberries, every type of lettuce you can think of, every type of herb you can think of, basil, chives. I mean, it was really a big productive system. While we didn't try to, it wasn't so much built as a specific scientific test in anthroponics, it was more to see, can it work and how much can we produce from it? So from then on, I went back to Portugal to finish my master thesis, and then I returned to Sweden at Hemmaodlat. And then I discussed with Niklas, maybe we can do some more experiments to see if we can figure out the specifics on how we can grow using urine. So first came our first experiment here. The objective was first to calculate how much does it really take for urine to go from its normal condition, when we just peed, and then to the condition where it's technically sterile, where the pH has increased to 9. At the same time, we would then use that pee to fertilize 3 different systems with 3 different urine dosages. One more information about the urine collection for the analysis of the pH. We did 3 batches of collection, each batch having 3 different jars, and all with the same volume, and all from the same urination event, as you can call it. So these systems were constant flood. They used the media. We used leca. They lasted for about, the whole experiment lasted for 2 months, although the actual growing part was only 1 month. And here you can see the evolution. From the first picture to the last, it only took 1 month. So it was a pretty big growth. Unfortunately for us, we wanted to test the different amounts of urine, but before we did that, we added a small amount of urine to cycle the system, that is, to increase the bacteria amount. That amount turned out to be enough just to grow the whole lettuce, which we were not expecting. So it was good and bad in the sense that we were surprised. So here are some of the results that we had. As you can see, the pH might take very different times to reach that pH level of 9. We did conclude that it takes about 4 to 5 weeks, just a little over a month, which is quite a long time to wait for something like this. And it would further inspire us to do another experiment later on. We did manage to grow about 626 grams of lettuce on those bags over there from 300 milliliters of urine. That's about the same amount of a can here. So urine is a powerful fertilizer, we found. We already had a hint about that, but now it was confirmed. We did some math as well, and we figured that an average human pees about or urinates about 1.4 liters of urine a day. So that means that every day one person could produce 3 kilos of lettuce, which is more lettuce than you need for a month or even more. And that was it, pretty much. This was our first experiment. Because it was a failed experiment in the sense that our initial objective was to measure how the plants would react to different amounts of urine, but we couldn't measure that, we decided to do a second experiment. And this time we decided to choose a plant that was more demanding in terms of nutrients. This plant was cucumber. So with the same system, in the first system we had about 108 milliliters of urine per week added. In the second system we had half of that, 54 milliliters, and the third one we had the double of the initial system, 200 milliliters per week. We only run the experiment for 1 month, because we didn't want to try and grow the whole cucumber. We just wanted to see how the different dosages would affect the leaves. And again, this is 1 month difference between the first picture and the second picture. But more importantly, in this other picture, you can see how the different systems grew differently. So from left to right, we have the system with the most amount of urine. So bigger growth, more as well green, vibrant colors. The system with the least amount of urine, the middle one, has the shortest growth. And finally, the other one had also a good growth. We also concluded that we shouldn't have added probably 2 cucumber plants, because we found out that one cucumber plant always overdeveloped over the other, which means it was stealing nutrients from one plant to the other. So this was something that we wanted to keep in mind for another future experiment. And here in more detail you can see how the different nutrient deficiencies affected the leaves of the plants. So you can see here that we have some signs of potassium deficiency, magnesium deficiency, and iron deficiency. We did achieve flowering, but cucumber plants tend to flower usually when they're also stressed. So this could also be a sign that we really were not supplying enough nutrients to all of them. To the more analytical side, we measured the pH over time. We saw that all pH decreased over time because of the nitrification process that occurred. However, the system with the most amount of urine decreased the most, which makes sense and is what we were expecting from the beginning of the experiment. We also measured the amount of nitrates, which tended to accumulate in all systems to the point where they were out of range of what we could measure. And again, the system with the most amount of urine added had the most nitrates accumulating. And the same with EC, or electroconductivity. The amount of salts just increased over time, but increased more in the system with the most amount of urine. So we took the values of some nutrients that we measured, potassium, magnesium, and iron, and then we compared it with some literature standard, for example, for growing hydroponics, for growing cucumbers in a hydroponic setting. So we found out that we had some deficiencies, and we started to think of how we could supplement these in the future, which will be the topic of a future slide. But more importantly, we figured that the ideal dosage of urine for a big feeder plant like cucumber would be somewhere between 109 milliliters per week to 218 milliliters per week. So before I mentioned that more than 1 month to wait for urine to reach that level of pH is quite a long time to wait. And if we want to bring this to bigger applications, we should find a way to reduce that time. So, and again, how can we speed up this aging process, the aging of urine, which increases the pH? So we decided to try watermelon seeds. Now, why do we choose watermelon seeds? When I was thinking about how we could decrease the time, I started looking at this equation about how urea transforms into ammonia more carefully. And I noticed something there, which was this enzyme called urease. Urease is an enzyme that takes urea on one hand and transforms it as much as there is around in the environment to carbamic acid and then to ammonia. Turns out the watermelon seeds contain urease, and they're a very cheap and natural source of it. All we had to do really was to dehusk the seeds, grind them down, and then add them to the urine. So I set out to make just that in a small experiment. What I found was very surprising. I had 3 jars. One jar had just the urine. The other jar had 1 gram of these seeds added, and the other had 10 grams. In the one that I added 10 grams, the effect was instantaneous. The pH just rose to 9 or more than 9 almost instantly, which was very surprising. And the other one took 20 minutes. Yeah, that's what's there. So 20 minutes. So this was really big breakthrough. This means that if we could manipulate the amount of seeds that we add, we can basically control when we want the urine to be ready to add. So on that topic, I decided to add just 1 seed of watermelon to just 1 jar, measure it over a short period of time, and then plot, as you can see here in this graph, over time, in a small period of time, how the pH changes. And then from there, you get an equation, and then you can calculate when do you expect the pH to be 9. And sure enough, after a time waiting, I went and checked the pH again, and it had rose to the expected level. So this was big news. However, the amount of jars that we did was not very good in terms of scientific consensus. It was not. The sample size was very small, so we really need to do this over and over again in a more controlled environment if we want to confirm this at an academic level. But for our intents and purposes, this was more than enough confirmation that we now can control how short of a time we need. So with that in mind, we started our 4th and ongoing experiment, which is how do we supplement this urine system, this anthroponic system, with a new source of fertilizer. In this system, we already used what we learned from the previous experiments. We prepared the urine and we added the watermelon seeds, and this managed us not to have to wait 1 month for the urine to be ready. So as you can see here in the picture, in one system we didn't add any wood ash. In another system we added a certain amount of wood ash, about 30 grams, and in the other we added about 70 grams.
Why did you add wood ash?
Exactly, I was getting to that. So it turns out that wood ash is a source of potassium that we noticed we had lacking in our previous experiment with the cucumbers. On the topic of that, it also had some other nutrients present in the ash that could supplement whatever was lacking. From our initial run of the experiment, you can see that indeed the cucumber plant looks more healthy the more wood ash is concerned. However, there is a big problem when we add wood ash, which is the pH. Wood ash is very alkaline, and when we add that to the system, the pH increased dramatically to the point where the plants became very stressed. So if one is to supplement a system with wood ash, we would have to add something to counterbalance the pH increase, for example, using phosphoric acid. So all of these experiments manage us to reach a certain amount of challenges we have to overcome so that we can bring anthroponics more into the playing field of growing. The first one is hormones and drugs. Like I mentioned before, even in conventional wastewater treatment, it's a hard thing to handle. Most wastewater treatment systems are not equipped for dealing with this. So in that regard, that challenge, we have to invest more in researching wastewater treatment at an academic level and a business level before we can take any conclusions and apply them to our systems. And then there's the odor and handling, which was most affected me because I was the one handling the urine. However, we can pretty much automate that in the future, and having either growing directly in the place of the collection, for example, having a toilet that has the plants growing in the back, or having a more sealed container, and then we can just take the nutrient solution after it's been filtered where the plants are growing. And then there's the perception. Nobody really would like to try urine-grown food if they were told they were urine-grown. Unless there were to be a big environmental problem and there is no food, then people would just not accept this at the moment. On the other hand, there's also no environmental laws or European laws that allow us to sell to customers this even if they wanted to. So there's still a big challenge in bringing this and proving that it works, that it's safe, and that people can consume it if they want. Another way to change this perception might be to having blind tasting experiments, where we have different types of a certain crop grown in different ways, and people can really taste and see the difference if there is any. Also, something that we haven't been able to test so much is the bacterial analysis on the surface of the plants or of the water itself, which might be something interesting to further prove or disprove that it is a safe growing method. Regarding nutrient deficiencies, as I mentioned before, we have wood ash as a potential source. We might have other sources that we are looking into. As always, something that is local should be privileged over something that is far away or requires a great transportation logistics to bring to market. And finally, could we integrate both wastewater to grow safely food for consumption? There are already some decentralized wastewater treatment technologies, but none of them include growing plants for consumption. So again, more research is needed in this area. And as for the future, we are planning on building a small anthroponics filter that acts the same way as I've described for these past experiments, but is contained in that the plants are not growing in the filter itself, but the filter does all of the necessary chemistry work for us, and then we can take the water from there and add it directly into a greenhouse, for example. So in this sense, we want to integrate anthroponics more in an urban environment so that people can grow their own food if they want using this method, and it's a safe method. And this could be, for example, added in office buildings or public buildings. The main question being in this system that there can't be any risk of contamination. There can't be someone that might be infected or healthy or unhealthy or under some medication that might change or destroy the system by adding this variable. So if we are to have this system for now, and because we're lacking some research on this field, we have to restrict what sorts of people can access this, what sorts of people can contribute to the urine, and how it grows. So that's it. If someone has any questions, feel free to shoot, to kill.
Did you see any deficiencies with the lettuce in the start?
Yeah, very little deficiencies, but there were some, yes.
Were they similar to the ones you saw in the cucumber?
Perhaps not at the same intensity, but they were similar. I would guess the most important one with the lettuce was the potassium. Yeah.
What did Alice think of your research? Did she part in that progress?
Could you repeat? Sorry.
The Alice?
Yeah.
Maybe?
Yes.
You were growing at her place?
No, she was doing it by herself.
Okay, but she had a part of it somehow?
Well, I've always asked her for advice, so I always contacted her.
But she had never done that before?
No, Before the one I described, no.
She thought it was interesting.
Yeah, yeah. She thought, why not?
But you said first that the male pea was better than the female.
It's not so much that it's better; it's that there is a risk that the female might. But
she
it's the
did
same.
care,
Yeah.
But she didn't take into account that.
No, what she described in the blog post that she was doing was that she took special care not to be under any medication and not to be, for example, with a cold or something. So she tried to make sure that everything was safe in her well-being, and then she collected the urine, and then she did the tests.
Isn't
So
it more easy with urine then? Poo, I mean.
what do you mean?
Because it has been for a long time ago, like for these solar survival earthship houses,
Hmm.
they poo in glass baskets, and it's very healthy, and they can use it to fertilize with.
I'm not familiar with that, but I would guess that
Because you mentioned
feces.
also poo,
Yes, feces has a very complicated problem, which is it has much more bacteria than pee.
yeah, but they
and-
say it disappear if you
Yeah,
some days it
Might be
in a
possible.
glass box.
might be possible. Yeah, but the one reason that feces is hard to handle, other than the odor and all of that, is also that it has a lot of vectors for contamination. There's a lot of viruses that we have in our intestinal system that get transmitted to that, and that's why it requires more treatment than urine would. So, and in some of the cases of urine where there might be contamination, sometimes it's also because it can become in close contact with feces. So, yeah,
That's the toughest maybe.
yeah,
And,
it would take some while for us to figure out a good way to do it. And I'm not sure Nicholas is keen on trying it here. I'm not keen on trying it. I'm scared.
We're
So,
not doing that.
yeah.
I was just thinking with the transformation of urea to ammonia
Hmm.
and volatilization with the pH. I was thinking when we add the wood ash
Hmm.
and increase the pH that way, would you think that adding the urine in an unsterilized form would help break down the potential hazardous bacteria that could exist because it's already in a high pH state, or is it another process?
I think it's a different process, but
Yeah.
it's always something that we can try out. From what I understand from the urea volatilization to ammonia, it's the chemical reaction itself that releases the ions that increases the pH of the urine. Whereas in the wood ash, we already have those ions there, and as soon as they come in contact with the water, they just increase the alkalinity. So I think there might be different processes going on, but it could be that.
Did you use any special type of biofilter for that? Or something special?
so-
Because it's urine.
No, when we use biofilter, we use something that, for example, many aquaponics people use, which is leca, or expanded clay pebbles. And by adding the source of ammonia, in this case urine, the bacteria develop over time there, and then they develop to grow and to deal with the amount that we add continuously. So it's not very special. We want something that is inert, durable, and has a high surface area so that we can have lots of little bacteria that convert that. So it's something that someone could do at home if they were keen on it.
So what you're saying, according to that circle diagram there,
Yeah.
is that if you pee in a jar, it's 95% water.
Yep.
Then you have like 2.5% minerals.
Yep.
Some sort of ions, and then you have this organic substance called urea,
Yep.
which is what? So that's not urine.
No.
So everything collected.
Urine
is
is all of it.
Yeah.
It's the name we give to all of this combined.
So urea is just some sort of organic molecule.
Yep.
And as soon as it reacts with the air, it starts to break down. Is that it? How does the reaction start?
The reaction starts by coming in contact with the air, but also with the water itself, which is part of it. So because there is a lot of water, it will quickly react.
That's
What?
Stew stuff or something? Egg protein?
Egg
Urea? Egg
Ah.
protein.
I'm not sure.
And it's also that one that gives the smell, the egg protein.
I don't know. But I do know that in some farmer fields or some farmers that use traditional soil, they buy urea in a solid form and then they sprinkle it over the fields. So this urea can be synthesized chemically in a laboratory, or it can come from us.
What's the purpose of adding urea and not say, is it a cheap source of nitrogen?
Yes.
Yeah. Okay.
So it's a main source of nitrogen.
Okay. So it's an organic source of nitrogen, and then when it reacts with the soil, it breaks down to ammonia, and then the
Yeah.
nitrification takes place.
Yeah.
Okay. That's interesting.
Yeah. But this should further reinforce that this small amount of minerals are what's powering that big production of lettuce, for example, that we saw, or even the cucumbers. Despite being highly diluted, it's still enough to produce a lot.
Do you see any other potential risks with using urine?
No, I would be more concerned about the supplements that we add, for example, using wood ash. Sometimes wood ash can come from a normal fireplace where metals are added, or ink from newspaper, and that can be uptaken by the plant. So that is a concern that we should address.
Well, with heavy metals.
Yes.
With cadmium, for instance.
Yes. So, for example, if someone consumes a crop that might have a heavy metal accumulated, that will maybe go into the urine and then affect us even worse. So.
And from the wood ash as well.
Yeah.
Because the tree takes up heavy metals. And
Yeah.
we burn it and then we add it.
Yes, yes.
So what you're saying here is, when we add the urine, we're adding something that is basic, that has a high alkalinity, because it's volatilized.
No, actually, urine is usually acid. So the pH of urine usually is between 6.5 and
After we
7.
add the melon seed, doesn't it, the pH raise?
After we add the?
Melon seeds?
Yes.
Yeah.
Then it
goes up.
raises through the process, not because the seeds themselves contain any.
Does it go down after the process is done, or does it stay high?
It stays high.
So when you add that to the water, does that affect the pH of the water?
The urine, you mean with the converted?
Exactly.
Yes, it does increase slightly the pH of the water. Over time, the nitrification will bring that pH down. But if we don't manage that pH, and if we just add it and let it go, the plant might become stressed by this suddenly high pH. So that's something we have to manage.
So what you're saying is you're adding a base in the form of the treated urine,
Yes.
which is now ammonia basically.
Yeah.
And the base in the form of wood ash.
Yes.
So, and that's the main problem. You have two bases that is.
Yep, that is the main problem. So what we might want to try, for example, someday is to find an equally cheap and local source of nutrient supplementation that is acid. And then by adding that, we can combat the high alkalinity of the wood ash and supplement all the nutrients that are needed.
Do you have any idea what that could be?
Not yet.
I don't either.
But for example, when we add phosphoric acid, we are essentially adding a type of some phosphorus into the system. It might take a while to be broken down, but we are supplementing a bit of phosphorus. So that's one way, but it's not exactly cheap, because the ones that we use here, for example, we call it pH down, and it's quite an expensive source of phosphoric acid. Yeah. Any more questions? Too much urine talk for today?
Super. Well, thank you so much.
Very interesting.
Thank you.
No problem.